Case Study Challenge, Spring 2018, by Emily Glynn, Sarah Patton, and Jessica Verhey. Meet Dixie. She's a purebred paint horse foal born just this morning. Dixie had an easy birth with no complications. Afterward, Dixie's mother appeared to be perfectly healthy. However, Dixie was not as lucky. Bum, bum, bum. It was soon discovered that Dixie was completely unable to stand. She also displayed further symptoms, including low energy, contracted tendons in her legs. Within 2 days, Dixie passed away. What went wrong? Glycogen branching enzyme deficiency, or GBED, is a condition in which the body is unable to store sugar properly. This results in foals dying young, stillbirths, and even aborted fetuses. GBED by the numbers. 3% of American quarter horses are heterozygous for GBED. The mutation of GBED is carried by 10% of all American quarter horses and related breeds, like the paint horse. GBED on the molecular level. Glucose isn't being converted to glycogen, causing there to be glucose in the muscles, which makes the muscles contract and not work properly. Glycogenesis is unable to occur. GBED is caused by a point mutation in the glycogen branching enzyme within the nucleotide sequence. The genetic disorder has been linked to the foundation quarter horse stallion, King. Symptoms include low energy, weakness and difficulty rising, low body temperature, contracted tendons in the legs, seizures, heart failure, low white blood cell count, and sudden death. On the right, we can see normal muscle, and on the far right, we can see muscle that has been affected by GBED. This condition can be diagnosed with a muscle biopsy from an affected animal, but the animal has to be deceased in order to obtain this muscle for the biopsy. Because GBED is recessive, parents of the foals must be carriers of the gene. There is no treatment. Genetic testing is available, and bloodlines can be examined to avoid the condition. Treatment could include finding the enzyme and injecting it into the muscle so that the receptor could continue the nucleotide sequence. This is the absent enzyme in the nucleotide sequence that causes glycogen enzyme branching deficiency. Without this enzyme, the horse's body is unable to convert glucose into glycogen. This results in the inability to store sugar, meaning that the horse will not have the proper fuel for their vital organs, muscles, and brain.